Hello, welcome back to Polymeric Biomaterials. We are now looking at the theme of biomedical polymers, which we have started looking at in the last couple of lectures, where we had a chance to see synthetic biomedical polymers in very great detail. And in this lecture and in the next lecture, we will turn our attention to natural biomedical polymers. And as part of this lecture, we will first take a little bit of time to recapitulate some key aspects of natural polymers that we have seen earlier, and then move on to discussing one very important specific class of natural biomedical polymers, namely proteins. And as part of protein structure property relationships, we will be primarily focusing on the hierarchical nature of protein structure and how that leads to specific properties and how that eventually impacts protein function in many different applications. So, as part of that, we will look at primary structure of proteins followed by secondary structure. Then we will see some aspects such as the presence of rotatable bonds, dihedral angles, and interactions that lead to protein conformations, all of which together jointly result in higher order protein structures, namely tertiary and quaternary structures. And towards the end of this lecture, we will look at structure property relations in the context of proteins by taking three very specific examples. One is collagen, which as we will see is a case of helices, silk fibroin, which as we will see is a case of sheets, beta sheets, and elastin, which as we will have a chance to see are natural elastomers. So, with that being the overview for today's lecture, we will now jump right into recapitulating first some aspects of natural biomedical polymers that we have seen earlier. So, this list is familiar to us. We have seen both merits, which are listed in green here, and perhaps some limitations of natural polymers. We have seen that many natural polymers can be considered bioactive because they have motifs for biological interactions. These are perhaps sequences that could be recognized by cells, for example, which can help cells attach to specific surfaces. Many of them are also biodegradable because they are found naturally, and hence the expectation is that when they are implanted eventually or used in a clinical application, they will be remodeled and degraded by the body without any concerns for what the degradation products could be. Typically, they have very minimal adverse side effects, especially if used in the pure form, if processed well and if sterilized correctly. They typically have very minimal adverse effects, which means they are cytocompatible. And because they are a renewable resource, we have a wide variety of options for choosing these kinds of polymers based on the application. And one example given here is type 1 collagen for any bone related application, because type 1 collagen in itself is a major component of bone tissue. That is the logic. Of course, there are some limitations vis-a-vis synthetic biomedical polymers. One of course is batch to batch variability that you will see in any of these polymers, natural polymers, and stability during both storage and processing. So, these are labile or sensitive biomolecules which makes them, or biopolymers, which makes them somewhat susceptible to degradation and changes both in structure and in function if not processed and stored correctly. So, special care has to be taken for many of them to ensure that they retain their structure which will eventually dictate the property and function. They can also undergo rapid degradation. Compared to synthetic biomedical polymers, natural biopolymers are not specifically known for their mechanical properties. There are always exceptions, as we will see. For example, silk is a very good exception. It is a biomedical polymer that is naturally derived, that has extremely good mechanical properties, but more generally speaking, natural biomedical polymers do not perform as well mechanically as synthetic biomedical polymers. Of course, there is difficulty in processing because many of them have charged groups, many of them have conformations, as we will see in proteins, that have to be retained. If the conformation of the protein is lost, then the function of the protein is lost. So, care has to be taken during processing and to ensure that they are also stable during processing. So, these are some aspects of natural biomedical polymers that we have had a chance to see earlier. Now, with that context being set, we will now turn our attention to the classification of natural polymers, and it so happens that they can broadly be classified into three broad categories. One is polypeptides, the other is polysaccharides, and the third is polynucleotides. And notice the word poly used in all three of them. This is just to reiterate the fact that we are still looking at polymers. So, the word poly there should indicate that many of the concepts we have seen earlier for polymeric systems will apply quite well to these kinds of polymers as well. Under polypeptides or proteins, we list a few types: collagen, silk and elastin, some of which we will have a chance to look at in this lecture. Under polysaccharides, we list again some: cellulose, chitosan, alginate with varied properties, which we will have a chance to look at in the subsequent lecture. And polynucleotides, these are largely based on DNA and RNA. This is not something we will look at in any detail in this course, but the hope is that if you are able to understand the processing, structure, property, function relationships in the context of proteins and polysaccharides, you can always find a way to apply some of those principles when you come across other kinds of polymers based on natural sources such as polynucleotides. There are two other examples listed here. Again, these are classes which are italicized. That is because those are more specialized, and there are many such types of biomacromolecules, two of which we are listing here. Glycosaminoglycans, or GAGs as they are called, and proteoglycans. These are specialized biomolecules that perform very specific functions, whose function is very directly related to the presence of some types of structure property relationships in these molecules. Glycosaminoglycans, for example, are very long linear polysaccharide chains that are highly negatively charged, and the reason why they are listed along with proteoglycans is because proteoglycans themselves are a combination of a core protein to which these long linear negatively charged GAGs are attached. So, that is what constitutes a proteoglycan. So, these are some specialized classes that have features of both polysaccharides, if we are talking about glycosaminoglycans, and proteoglycans, which also have a protein core. So, as you can see, we can have many such types of biomacromolecules. Of course, this is again a very small subset, but the hope is that if you can understand the principles and the concepts involved in how hierarchy prevails in some of these systems, then you can easily appreciate these principles when you come across them in the context of other biopolymers in a later course. So, with that being said, we will now start looking at proteins in more detail. We have talked about the importance of hierarchy in polymeric biomaterials quite a bit early on in the course, and in my opinion, proteins are a beautiful way for us to visualize what hierarchy really means in polymeric biomaterials. Because proteins naturally have what you can think of as at least 3 or 4 levels of structure depending on the protein. Broadly known as the primary structure, indicated sometimes by this. One level higher would be the second order or secondary structure, indicated by this. And then when protein folds, it can have its own conformation, and that is the tertiary structure, indicated by this. And some proteins have multiple chains that are joined together to result in quaternary structures. So, there are at least 3 if not 4 levels of protein structures that we can talk about in terms of hierarchical organization, and all of them are important for different reasons, as we will see very shortly. So, this is a good example of where structure directly impacts property, which impacts the function of the protein. Now, if you think of proteins as polypeptides, one immediate question you can ask is, what is the monomer used for protein synthesis? Now, in lecture 11, we looked at polymer synthesis or polymerization in great detail, but our focus largely was on synthetic polymers. Here, we are now looking at natural polymers in the specific case of proteins, and we see that for a protein or a polypeptide, the starting monomer is what you would call an amino acid. A generic version of that is shown here, where you have this carbon in the middle, which is called the alpha carbon, which is attached to the amine on one side, the carboxylic acid on one side. So, that should explain the name amino acid right away. So, you see how intuitive it is. And then you also have a hydrogen attached to it, and for the fourth bond we are showing a generic R group, which will change based on the nature of the amino acid. And the distinguishing feature from one amino acid to the other is the R group. In one it could be just hydrogen. For example, in glycine the R group is just a hydrogen. In alanine it's something else, in lysine it's something else, and we will have a chance to look at some examples of these R groups and how they directly contribute to the properties of the amino acid. But just to keep in mind that this is the starting point of the monomer of the protein. Also, one thing you will immediately notice is that the alpha carbon is a chiral center. So, except for glycine, where the R group also happens to be a hydrogen, all the other amino acids can exist in the L or the D isomer forms. And we have seen what chirality is, we have seen what stereoisomers are, what enantiomers are in some detail in lecture 7. So, now would be a good time to pause and recall some of those concepts that we have seen earlier. And in nature largely you will typically find L amino acids. So, when people refer to L or D, they are referring to the isomer, either L or the D isomer, and largely our discussion is going to focus on the L isomer because that is what is mostly found in nature. Now, shown here is a cartoon or a schematic of all four levels of protein structure, and while we will look at each of them in more detail in subsequent slides, this is a good snapshot of the different levels of protein organization based on the nature of interactions. So, the first thing you will notice is that the primary structure, which is simply the amino acid sequence as shown here, the primary bond that we are talking about here is the covalent bond that links one monomer to another. This is not unlike any other polymer we have seen in the course so far, where the primary criterion that we saw for the backbone was covalent bonds that link the carbon atoms together, for example. And then when the protein starts exhibiting secondary structures or higher orders structures, two examples of which are shown here, beta sheet and alpha helix. The primary bonds that we are talking about there are hydrogen bonds, which stabilize the secondary structures. And proteins, like we said, can also take up tertiary structures, and shown here is one example of one such protein, where the bonds could be quite varied and diverse. Largely we are talking about non-covalent bonds here, such as electrostatic interactions, hydrogen bonding, hydrophobic interactions, van der Waals forces. You can also have disulfide bonds which are covalent, and that is the exception, and we will look at which amino acid largely contributes to that in just a while. But this is just to give you an idea of what types of interactions govern the different hierarchical levels of organization for protein structure. And of course, like we said, some proteins that have multiple subunits, multiple polypeptide chains which are linked together, the links could be non-covalent interactions or could be disulfide bonds. Again, just to show in one picture, one schematic, how to visualize or think of protein structure at the primary, secondary, tertiary and quaternary levels based on the nature of interactions. And as we will see, it turns out that these different interactions are super important for how a protein folds in three-dimensional space and how it eventually functions and does its job as it's expected to do. So, we will first begin looking at the details of the primary structure of proteins. So, these are, if you think of it from a polymerization perspective, these are enzyme catalyzed condensation reactions. Now, condensation should immediately bring up an image of a reaction where water is eliminated. That is something that we have seen in lecture 11. So, here something very similar happens. You have two amino acids, one shown here, second one here, and when the two of them combine, water is eliminated, resulting in what we call the amide or the peptide bond. The CONH, which is the amide linkage, is also called the peptide bond. So, this is very much a covalent linkage, and this is what holds the chain together. Right? So, one amino acid is connected to the other, is connected to the other, is connected to the other, and so on till the end of the protein, and those interactions are covalent, connected by this amide or peptide bond. So, the primary structure of the protein is simply the linear ordering of the L amino acids linked between each other by the amide or the peptide bonds. Now, for proteins, the directionality becomes very important. If you now extend this argument for many different amino acids, you will very quickly see that once the full protein is made, once all the amino acids are linked together by covalent bonds, you will end up with one free amine on one side and one free carboxylic acid on the other side. So, this is the first amino acid whose amine will stick out this side. This is the last amino acid whose carboxylic acid will stick out on the other side, which are not involved in the amide bonds. And this is called the N terminus, this is called the C terminus. And by convention, protein structure in the primary form is written from N to C, N terminus to C terminus. This is a very important point to note. This directionality is very important because you can have the same set of amino acids, and if the ordering is different or if the arrangement is from C to N, you will get very different properties compared to N to C. So, whenever you list out the structure, you have to be very clear about what is the ordering of the amino acids from the N terminal to the C terminal. So, that is what we are saying here. There is the N terminus where the free amine group is there. There is the C terminus on the other end of the chain. So, you have to imagine that the chain, polypeptide chain, goes all the way from one side to the other, and you have the carboxylic group on the other side. Now, if you have to think about how long is the protein, there are a few different ways to think about it, and just as for polymers, you can also think about a degree of polymerization for proteins or polypeptides. So, you can have anywhere from 50, 100 amino acids all the way up to 2000 amino acids in many cases, or even higher for proteins. So, that is sort of the range you are talking about. Those are the kinds of numbers you are talking about for many commonly seen proteins, say about 50, 100 amino acids linked together by covalent bonds all the way up to 1500, 2000 amino acids. You can also think about molar mass for proteins, and as we have seen earlier, proteins are monodisperse systems in that when you say, for example, bovine serum albumin is a very popular protein. Whose molar mass is fixed, and that is because the amino acids in BSA, this is short, in short it is called BSA, the amino acids for BSA are fixed, and not only are the amino acids fixed, the ordering of the amino acids are also fixed. Now, this is quite different from, let us say, polyethylene, where we have seen in this course itself, we have seen different versions of polyethylene, from low density, high density, ultra high molecular weight polyethylene. So, we have seen a range of different types of polyethylene, all with different molar masses, perhaps even different polydispersities depending on the synthesis and so on. So, this is one distinguishing feature of proteins where the systems are monodisperse, the molar mass is fixed. Now, just to drive home the point about why both the amino acid type and the order are important, sickle cell anemia is a condition that is quite widely seen in certain populations, and there the underlying cause for why this happens, where the red blood cells take on a different shape compared to the healthy population. Believe it or not, it is just a single amino acid substitution that has gone rogue. So, valine for glutamine in hemoglobin, that substitution leads to this condition called sickle cell anemia. So, just that one amino acid substitution makes such a large difference, right. So, just to keep in mind that not just the amino acid type, but the ordering is also very important. Now, we have hinted earlier that the amino acids are distinguished by the R group. So, now would be a good time for us to look at different types of R groups that can possibly exist in amino acids and what their distinguishing features are. So, the first thing to note is almost all amino acids, most of them are zwitterions at physiological pH, and zwitterions simply is a molecule where you have both positive and negative charge on the same system, but the overall net charge will be 0. And for an amino acid, it is not very hard to imagine that the NH2 can get protonated. It is a good proton acceptor, so it can become NH3 plus, and then if you have the hydrogen here and the R group here, likewise the carboxylic acid can donate the proton and become deprotonated to become COO minus. So, you see, this is a good example of a zwitterion where you have both the positive and the negative charge on the same molecule, but the overall net charge is 0. One important point to also note is that the R group can also have charge. So, a related concept that you will see when you look at papers, when you look at protein properties, is this idea called isoelectric point. So, when you take a protein, so what we saw was just for one amino acid. Now, for a protein, you will have many hundreds of amino acids, each having R groups that may or may not have charge, and overall there could be a charge on the protein at physiological pH. Now, isoelectric point is that pH at which the net charge on the protein becomes 0. So, that is a distinguishing characteristic feature for every protein. For a given protein, you can define an isoelectric point where it is the pH at which the net charge. So, you can still have charges throughout the protein, but the overall charge on the protein is 0. So, in the presence of an electric field, it will not show any migration when it is reached that pH. That is how the isoelectric point is defined. So, that is an important characteristic feature of proteins that you will come across. So, you have already seen number of amino acids, the ordering of amino acids, molar mass and isoelectric points. These are all important features of any protein that you can work with or will come across. So, when you come across a new protein, stop and ask maybe along these 4 categories, what are the properties of that particular protein. So, now coming back to the R group in the amino acids. Now, as we said earlier, there are 20 naturally found amino acids. Sometimes they are also called residues when they are in a protein. So, instead of mer, you remember the word mer that we used for polymers in general, the equivalent in protein literature would be a residue, and the amino acid itself would be the equivalent of a monomer. So, you can have 20, like I said, 20 amino acids naturally found, and one typically when you read, if you've taken any biochemistry course, Biochemistry 101, you will have very likely seen a chart that lists out the structures of all amino acids. And one thing that is often problematic for students is, should I remember the structures of all amino acids? What we are going to see now is a slightly more intuitive way to understand amino acid structure, and the goal is not so much to remember the structure of the R group of every amino acid, because that's something you can always look up in a textbook if you need, but to look at it from the perspective of what possible interactions can that amino acid have along maybe some 4 or 5 categories. The first category we will look at is a group of amino acids which have a non-polar aliphatic R group. So, the R groups in all these amino acids are highlighted in yellow, and what you will see for this amino acid shown here, valine, is this group here is aliphatic, there is no aromatic ring there, and it is non-polar. That should be obvious just looking at the methyl groups. What you will also see is there are at least 3 ways to designate amino acids. One is of course the full name of the amino acid, in this case it is valine. The other is a 3-letter abbreviation, VAL in this case, or a 1-letter abbreviation which is V. So, very often you will find when protein primary structures are written, either the 3-letter or the single letter abbreviations are used. You can also have non-polar aromatic R group. So, this is still non-polar, but you have a ring. So, it becomes a aromatic R group which is non-polar, and this happens to be this specific amino acid called phenylalanine, which is abbreviated as PHE or F. Another category that is equally important is a polar but uncharged R group, and shown here is an example of serine, which is abbreviated as SER or S, and you see here that the OH group makes it polar without the explicit presence of charge. So, those 3 on top are not charged, and what you have on the bottom, these 2 are the charged amino acids. Obviously, one would be positively charged, you will have a negatively charged category also. So, shown here for under the category of the positively charged R group, you have the NH3 plus which is lysine along this R group linkage, and for aspartate you have this COO minus which is negatively charged. Sometimes it is called aspartic acid, in which case it will be written as COOH. So, this will be written as COOH. So, when it is aspartate, you see the explicit presence of the negative charge. So, what you see here from this description is more than remembering the structures or the R groups of all the amino acids. What is more important is for us to understand what kind of groups are we talking about here, and under each we have looked at simply one example. We have more examples under this category, we have more examples under this category, this and this. But simply based on the examples, one example that we have seen under each category, the hope is that you will be able to appreciate what kinds of interactions these amino acids can participate in, which is what really determines how a protein can fold in 3-dimensional space. So, that is the connection we are going to make. So, before we look at the secondary structure of protein, one of the first questions we can ask is, why does the protein fold at all in the first place? Why isn't it that the primary linear chain good enough? Why does the protein have to fold at all? Now, protein folding is a topic on its own, but for the purposes of this discussion, a couple of things you will note. One is that as we just saw, there are both hydrophobic or non-polar amino acid residues and polar or charged amino acid residues. And because proteins exist in an aqueous environment surrounded by water, whether we like it or not, that is the medium found in the body, and proteins exist in an aqueous environment, it's energetically unfavorable for the hydrophobic or the non-polar amino acid R groups to interact with water. So, typically those get buried inside and want to expel the water from their interactions. This is more popularly called the hydrophobic effect. This is why oil does not mix with water, for example. So, as a result of the uncharged non-polar amino acid residues getting buried inside, the charged or more polar residues stick out and interact with the environment outside or with each other or with the water outside, and that is one reason why the protein folding happens. Protein folding also can be thought of from a thermodynamic perspective as an energy minimization problem. If you think of free energy, the protein along the chain wants to maximize its interactions, favorable interactions, and minimize its unfavorable interactions such that overall for that temperature and pressure, the protein in its lowest energy, free energy state, right. Also, one thing to note is we saw a lot of carbonyls and amines, right. So, we saw the CO, NH, the CO and the NH, both of them can participate in hydrogen bonding with water. But as a result of the hydrophobic residues being buried inside, some of these hydrogen bonding interactions with water are lost. So, the protein in many ways wants to compensate for that loss by forming hydrogen bonds through the formation of secondary structures. So, these are some ways to think about higher order structures in proteins, but like I said, protein folding thermodynamics is not a topic that we will get into in great detail in this course, except to note that these are some of the reasons why proteins like to fold in 3-dimensional space. So, with that we will segue nicely into the secondary structure of proteins, where the primary bonds we are talking about are hydrogen bonds between the carboxyl O and the amine H, which leads to twists and folds, largely resulting in two important classes of secondary structures, either a helix or a sheet. We will look at in more detail in just a bit, but we will note here that the propensity or the probability of a particular sequence of amino acids to form either a helix or a sheet depends on the exact nature of the sequence. So, you can first have the alpha helix, where the picture here shows what an alpha helix will look like, one part of it will look like. So, this is characterized as a right-handed helix. So, right-handed helix will have this kind of an orientation with 3.6 amino acids per turn, where all the R groups are oriented outside the helix. And one important feature of the alpha helix is that there is a hydrogen bond that forms between the carbonyl O of the ith residue and the amide H of the I plus 4th residue as shown here. So, this carbonyl O hydrogen bonds with this amine H. So, this black line, dotted line indicates the hydrogen bond, likewise here. This carbonyl O of this residue hydrogen bonds with the amine H of this particular residue, and so on and so forth. And note here that we have again listed the directionality from N to C. One thing to keep in mind though is, as the protein forms these localized secondary structures, some amino acids are not very well accommodated in a helix. This is why I said earlier that the propensity to form a helix or a sheet depends on the actual nature of amino acids in that sequence. For example, you can have steric hindrance. Some amino acids may offer steric hindrance such that they cannot fit very well within an alpha helix, or they can have hydrogen bond donor or acceptor too near the main chain, which will again cause difficulty in them being a part of the helix. Now, another type of secondary structure that proteins can form is what is called a beta sheet. a picture of which is shown here. And one thing to note here before we look at the beta sheet in more detail is, for both these cartoons, the hydrogens on the alpha carbon are omitted just for clarity, and the cartoons are slightly exaggerated to emphasize the hydrogen bond. So, because our goal is to understand protein structure from the perspective of interactions, we are primarily focusing on the hydrogen bonds. So, some of the bond angles etc. could be exaggerated. Notice here that in the beta sheet, unlike the alpha helix, the chains take up a linear zigzag kind of a conformation. So, the hydrogen bonding happens between the carbonyl O again and the amine H across chains as shown here, and you can have two varieties which you can immediately appreciate. You can have a chain that goes like this and goes like this, and you can have hydrogen bonds across the carbonyl O and the amine H across the chains, or you can have a chain that goes like this, makes a 180-degree turn, twists itself and comes like this, and you can have hydrogen bonding between these two. In the former case, that is called a parallel structure. In this case, the latter case, it is called an anti-parallel beta sheet structure, like so. So, shown here is an example of anti-parallel beta sheet where N to C goes this way, and on the bottom the chain reverses and comes back this way. And in the beta sheet, the R's on alternate amino acids point either up or down as shown here. Now, one way for the protein to make these turns is through what are called beta turns. These are short amino acid sequences, 4 residues, that allow the protein chain to make hairpin like bends and almost turn 180 degrees if you will, to fold back such that these hydrogen bonds are possible. Now, apart from alpha helices and beta sheets, you can have other types of secondary structures as well. Beta turns, as we have already seen. You can also have random coils. Now, when we say random coils, what that means is those are regions in the protein where there is no specific or regular structure or regularity of structure is missing. Those typically occur at the termini, the ends of the chain. You can also have them appearing in between two helices, for example, or two sheets, for example, or a sheet and a helix, for example, and those are the regions that contribute to protein flexibility. And as we will see later when we look at silk, the beta sheet like crystal structure will lead to rigidity in the protein. So, that is the secondary structure of proteins. Now, before we move on to tertiary structures, we will note a few more things about protein conformations which will help us explain how tertiary structures are formed. Now, for that we first look at we take a closer look at the backbone itself. What we are showing here is a segment or a part of the backbone where we have the alpha carbon here, and then we have this CO here, we have the CO here as well. These are both alpha carbons as well, and then we have the NH here and the NH here. And for us to understand how a protein can even fold, we have to first understand how the angles between the bonds can rotate. And this is not a new concept to us. We have seen this in very great detail in lecture 9 when we looked at the concept of dihedral angles. So, we will quickly go over how to contextualize that for peptides and proteins, but for more details I would encourage you to pause and recall some of these concepts from lecture 9. So, first of all, we note that dihedral angles are just angle made between two planes, and to define a dihedral angle you need 4 atoms or 3 bonds. So, you see here the first dihedral angle, which is phi, is involving Ci-1, Ni, Ci alpha and Ci. And this is the angle of rotation when you look through from that side. And the second dihedral angle, which is psi, involves Ni, Ci alpha, Ci and Ni plus 1, which is given as this. And note that the CONH bond, which is the peptide bond itself, given by omega, which is this bond or this bond as well. These are more rigid and planar. So, if you want to think of the protein, if you want to recall our familiar example of a noodle or a spaghetti, a cooked spaghetti, that is a good analogy to think of. Imagine that the bonds representing the peptide bond are slightly undercooked. So, they are stiffer, and the bonds representing these are fully cooked, which means they are more flexible. The bonds can rotate. In some sense, the peptide bond has a partial double bond like nature, which makes it not very easy to rotate, unlike the other single bonds that we are seeing in this example. So, why is this important? This is important because this is what allows a combination of rotations between this phi and psi. This is what allows the peptide or the polypeptide of the protein to sort of rotate along the chain such that you get different conformations. But as we have seen earlier, what limits the conformations and what kind of angle combinations they can take are energetic considerations, largely steric based on steric hindrance. So, not all angles will be favored for both phi and psi, and if you want to get a good sense of the combinations that are allowed for real proteins, I would encourage you to pause now and look at what we would call the Ramachandran plot. Now, this is named after Professor G.N. Ramachandran, who was a pioneer in studying the structure of proteins and peptides, and he was one of the first people to report the structure of collagen. So, this was the period in the 1950s, which was a very interesting, very important period for structural biology, where three very important structures were clarified in that period. One was Linus Pauling, who showed the existence of alpha helices and beta sheets, the secondary structure of proteins. And then Watson and Crick, who clarified the structure, double helix structure of DNA, and Professor Ramachandran, who showed evidence for the triple helical nature of the collagen molecule. Now, Pauling went on to win the Nobel Prize for some of his work, and Watson and Crick, along with Maurice Wilkins, also won the Nobel Prize. Notable omission in that group was Rosalind Franklin. And Ramachandran, G.N. Ramachandran also did not get the Nobel Prize, although many scientists would in that period and later have argued that they were probably strong contenders for the Nobel Prize as well. A lot of this was possible because of X-ray crystallography. And that period is a very interesting period in the history of scientific developments, because there is a lot of intense competition to who would crack the structures first. In fact, Linus Pauling himself had proposed an incorrect triple helical model for DNA until Watson and Crick, along with Wilkins, with data from Franklin, were able to crack it. And Ramachandran, of course, cracked the triple helical structure of collagen, and he also came up with this idea of looking at combinations of dihedral angles between, so if you look at a Ramachandran plot, you will see combinations that are allowed and disallowed So, it is a plot between this phi and psi, phi on one side, psi on the other, and the allowed regions will tell you if something can form a beta sheet or an alpha helix, and disallowed regions based on energetic considerations and steric hindrances will show that those, while in theory are possible, are not energetically or practically favored or thermodynamically not favored. So, some just quick trivia to get you excited about this point, and if you look up the Ramachandran plot, and maybe you can sort of trace your way back and look up the history of the developments in structural biology in the 1950s. It is a very interesting period in history. Coming back to now protein folding. So, put a pin on this idea, the fact that these bonds can rotate, these dihedral angles or torsional angles can rotate. That is one aspect to keep in mind. The other aspect to keep in mind for protein folding is the interactions. Now, just because they can rotate doesn't mean they will always rotate. One reason for them to rotate and lead to different protein conformations is also because certain interactions get maximized and favored, and certain interactions that are unfavorable get minimized in the interest of free energy minimization. So, shown here is in one chain sort of an exaggerated version, but in one chain we are trying to show some possible interactions that can exist within a chain, and this could also be across chains, if you will. So, you can have for example, van der Waals and hydrophobic interactions. So, this is what you would call hydrophobic interactions. You see all these valines here and here want to cluster together, expelling the water such that they can interact with each other in that cozy little space. You can also have hydrogen bond formation between a residue here and a residue here. Now, I will leave it as an exercise to you to sort of go back and look up amino acid sequences and see what kind of amino acids could have this kind of a R group that sticks out. It is an interesting exercise that I will leave it for you to look at. You can also have electrostatic interactions. We have seen that amino acids can also be polar and charged. So, if you have such amino acids across, you can have electrostatic interactions like so. You can of course have van der Waals interactions. These are broadly dispersion forces based on dipole moments, and the one exception here of course is cysteine, which can participate in covalent interactions in what is called a disulfide bridge. So, cysteine is an amino acid that is unique because it has a thiol group. Now, two cysteines, one here and one here, can interact and form a divalent disulfide bridge through a covalent bond based on the oxidation or the reduction state of the cysteine itself. So, in the oxidized state, they can form this disulfide bridge, and if in the presence of a reducing environment, they can go back to the SH form. An interesting trivia to sort of go back and for you to look up is there is an interesting connection between the presence of cysteine and its oxidation states and to the waviness or the curly nature of hair, human hair. The fact that some hair types are curled and some hair types are not, believe it or not, has a very interesting connection to cysteines and their oxidation states, something that you should perhaps look up. But just to summarize this slide, there are two aspects now that are now leading us from primary and secondary structures to higher order structures, which is the fact that there are dihedral angles that can rotate, thus giving the protein flexibility or the backbone flexibility, and specific interactions across different amino acids or the R groups across amino acids along the chain, which help the protein fold in very specific ways. So, both of these will lead to higher order structures. So, as we have said earlier, the charged and polar R groups typically map to protein surfaces. Because of the hydrophobic effect, the non-polar groups, R groups, tend to be buried in protein cores. What does this do to the protein? It leads to higher order structures. So, because of these interactions and the fact that the dihedral angles can rotate, the protein now starts to fold in three-dimensional space. Now, there is also a very important reason why the protein folds. In many cases, protein folding or the conformation in 3D is what determines the function of the protein. So, here is a direct link between structure, and when I say structure here, I mean the conformation of the protein in 3D to the function of the protein. And this folding is largely governed by interactions between distant amino acids on a chain. And as we have seen on the previous slide, these could be ionic interactions, hydrogen bonds, hydrophobic interactions, it could involve van der Waals forces, and of course, the exception is which is non-covalent, which is not non-covalent, is the cysteine-based covalent disulfide bridges. So that's the tertiary structure. Now, it's also not very difficult for you to think of the reverse process, which is protein denaturation. So, when we earlier said proteins have to be stored very carefully in protected environments, stored cold, etc., this is to ensure that the temperature effects or heat does not destroy or denature the protein. And when we say denature, can you figure out what level structure we are talking about for a protein? Are we talking about the primary structure or secondary structure or tertiary structure? One way to think about the answer to that question is to ask what types of bonds hold the different levels of structures together and which ones are the easiest to break from a bond energy perspective. Then you can easily arrive at the answer and see that it's the tertiary structure and secondary structures that can get disrupted somewhat much more easily than the primary structure, which is just a linear chain made up of covalent linkages which are very difficult to break. Now, in some cases, some proteins have multiple chains and they can be linked together by non-covalent bonds. So, in this case, when you have multiple chains, each chain in a protein subunit is called a monomer. So, you can have a dimer if there are 2 chains, you can have a trimer, you can have a tetramer if there are 4 polypeptide chains that are linked together. That is called the quaternary structure of the protein. Now, some manifestations of that include hemoglobin, where the quaternary structure helps or aids enhanced binding of oxygen, immunoglobulin, where multiple receptor responses are aided because of the quaternary structure, and actin, where again the quaternary structure helps give the cell shape and form. 3 very different functions, but what connects all of them is the fact that they have a 4th level of structure where multiple polypeptide chains come together and form as subunits and form one massive structure. So, one example that we will look at very quickly is this MTAP, which is just an enzyme, and the way you know it as an enzyme is it ends in ASE. Enzymes are proteins, and the reason I'm showing this particular example is because it has a quaternary structure, which is a trimer with 3 identical subunits shown here in blue, red and green. Now, in this ribbon model, you can possibly try and see if you can figure out where the alpha helices are and if you can perhaps see where the beta sheets are. The beta sheets typically are represented as arrows like this, whereas the alphas are typically represented as spring or coil like structures. So, that is one representation of the protein 3-dimensional structure. Now, the reason we are also looking at this particular protein is this quaternary structure conformation is very important to the functioning of this protein. So, the active site for this enzyme is located in the center where the 3 subunits form a torus like structure, and that is where the enzyme binds to the substrate like the MTA and facilitates the reaction. So, in one example we have seen the fact that a protein can take up a quaternary structure and how it is directly related to its functioning. Now that we have broadly looked at protein structures at multiple levels, we will quickly look at 3 specific proteins that are important from polymeric biomaterials perspective. The first of them is collagen, which is a structural protein. Now, there are many types of collagen found in the human body. Some of them take on the fibrillar form, some of them are non-fibrillar. The major types are collagen 1, 2 and 3. There are of course 4, 5, 10 etc. And structurally speaking, collagen has 3 left-handed polyproline type 2 helices. Now these are not alpha helices, they are also helices but not alpha helices. The key differences being that firstly these are left-handed helices as opposed to right-handed alpha helices. Second is in alpha helix we saw that the number of amino acid residues per turn is 3.6. Here it is more like 3.3. And then how exposed it is to the solvent etc. is different between the alpha helix and the left-handed polyproline type of a helix. And the fact that it is called a polyproline helix should indicate to you that there are proline rich regions. Proline is an amino acid and there are proline rich regions in the backbone. So collagen is one such protein. From a primary chain perspective, collagen chains contain multiple repeats of glycine and this XY kind of a repeat unit where X and Y are typically proline, hydroxyl proline or lysine and hydroxyl lysine. And proline and lysine are naturally found amino acids. We are showing simply the hydroxylated versions here where the hydroxyl groups are highlighted in yellow. Now 1/3 of this collagen molecule is glycine, and why this particular set of amino acids constitute collagen is also very interesting. There is a very specific reason from an evolutionary biology perspective. There is a very specific reason why it is this combination of amino acids that results in this specific structure for collagen. So firstly we note that glycine is the smallest amino acid. So it can easily fit in and it increases the flexibility, thus allowing the formation of these helix-like structures. Proline and hydroxyl proline, as you can see from the structure, provide structure because they are rigid, they provide rigidity, and lysine or hydroxyl lysine is what helps connect two chains of collagen through covalent crosslinks, and those crosslinks directly impact the strength of collagen. So you see, we are going from connecting why an amino acid should exist in this protein to how it will affect protein folding in the form of a helix to crosslinks across chains that results in bulk mechanical properties that you can measure. So you see how many length scales we have looked at in terms of structure property relations. So this is why I said proteins are an excellent example of biomolecules or molecules, macromolecules in general, where we can understand structure property relationships very well. And higher order structures in collagen typically include triple helical structure with the 3 chains. So the 3 left-handed polyproline type 2 helices held together both by hydrogen bonding between proline carboxyl carbonyl groups on one chain and the glycine NH groups on another chain, as well as water mediated hydrogen bonds. So shown here is a schematic of what the triple helix structure will look like, where you see 3 different colors. One is yellow, the other is cyan, the other is purple, and you see that each of those colors represent one distinct polyproline type 2 helix, and together they intertwine to form this triple helical structure of collagen. Typically you are talking about length scales in terms of width, you are talking about 1 to 2 nanometers and 300 nanometers long and 3.3 residues per turn, which is different from the 3.6 residues per turn that we saw in the case of alpha helix. Now this also leads to a hierarchical structure in collagen. So what you have here is the collagen fibril itself. If you want to think about the highest level of structure, of course you can have fibers on top where multiple fibrils stack together, and then if you zoom in on just one particular region, so this fibril is simply many triple helical chains cross-linked through the lysines, which we will see very quickly in just a bit. But if you zoom in into one region, you can have a single collagen molecule, where if you zoom in further, you will see that it is the collagen triple helix, If you zoom in further and you take out just one polypeptide chain, you can get this single chain, which is just the left-handed helix. And then if you zoom in further, you will see this glycine XY repeats. So you see, if you go in the reverse direction, the amino acid sequence results in the left-handed helix, and then 3 such chains, helices, intertwine together to form the triple helix, and then multiple triple helices cross-link together to form a fibril, and these crosslinks are covalent crosslinks across chains through the lysine groups. So that is what we say here, and that happens through the process of deamination of lysines or hydroxyl lysines by this enzyme called lysyl oxidase, where the formation of aldehydes is promoted, and then 2 aldehydes across the 2 chains. So you have one triple helix here, one triple helix here, you have 2 aldehydes sticking out. Through aldol condensation, they can cross-link. That results in very strong tensile properties for collagen. When we look at mechanical properties under bulk behavior and responses later in this course, we will again revisit collagen, and we will ask, as the collagen molecules get stretched, what happens to both the structure at the microscopic scale as well as the bulk. But we will note here that the strength of collagen on a per weight basis approaches that of steel, and one reason for this very strong tensile properties of collagen is because of the hierarchical structure, because of the crosslinks, and because of the triple helix with each of the helices being left-handed and all of them are intertwined. So, multiple reasons for why collagen derives its strength. And collagen is present in many tissues, and differences in mechanical properties across tissues have to do with how collagen reorients under strain and how water is removed, as well as other aspects of the matrix. So, collagen alone does not contribute. In the case of bone, for example, you will have collagen that is associated with the presence of hydroxyapatite like mineral, which also will contribute to mechanical properties. You can also have other associated proteins like elastin, which will also contribute to mechanical properties. And like I said, as we will see later, we will see that ligaments, for which collagens are a very important constituent, will show a triphasic response when you stretch them. When you conduct tensile testing on a ligament, you will see 3 distinct phases in the response, and we can attribute all 3 phases to specific structural changes that happen in the collagen molecule. And we will note here that collagen is popular as dermal fillers, and is also in FDA approved for spinal and orthopedic procedures as bone grafts in the form of infuse, which is simply collagen plus recombinant human bone morphogenic protein, which is another protein, it's a cytokine that induces bone formation. So, it is a combination that can help provide support and regeneration of bone in specific applications. Another example that we will look at now, looking at sheets, is silk fibroin. This is something we have looked at briefly in an earlier lecture. We will again reiterate some key concepts here. Now, what we are talking about here first, you have to note, is silk fibroin. There is also a sericin component to silk, which can be removed through this process called degumming. Sericin is removed through degumming, and we are primarily focusing on the fibroin component here, which is obtained from this silkworm. And there are 2 distinct chains, the heavy chains and the light chains, which are linked by disulfide bonds, and by now you should be able to tell which amino acid would be responsible for that. And the primary structure of the heavy chain largely consists of glycine, alanine and serine, with repetitive sequence contributing to strength of silk. Now, this repetitive sequence should ring a bell when we looked at crystallinity. In lecture 12, we saw that this was a key feature of something to note for crystalline materials. So, the fact that there is a repeating sequence or there is some order in the structure sort of indicates that we might be able to see the possibility of some crystal-like arrangements in silk. So, the high glycine and alanine content allows for very high tight packing, leading to very rigid structure and very high tensile strength, and shown here is just a comparison. So, silk, like I said at the beginning of this lecture, is an exception. Among natural polymers, it is one of the strongest polymers, and you see its properties far outperform some of the synthetic polymers themselves. So, the hydrophobic beta sheet crystallite domains, which largely comprise the glycine and alanine, are surrounded by the hydrophilic non-repetitive amorphous regions, namely tyrosine and serine, and shown here as a cartoon of what that microstructure will look like. And these are anti-parallel beta sheets, which would be considered the crystallites, which are largely held together, as we have seen earlier, through hydrogen bonding, and the amorphous regions confer flexibility and ductility. Now, there is one very interesting property about silk as well. Silk in its natural form is not soluble in water, but for processability, if you want to process this into interesting architectures, etc., for specific biomedical applications, water solubility is desirable. So, for that, what is typically done is an agent such as lithium bromide is used to disrupt all the hydrogen bonding interactions in silk, and then it goes through a series of processing steps thereafter, and then the regenerated form of silk that you obtain from that process will be water soluble. So, what you can do now is process that silk, and once you have processed it into the final shape and architecture that you want, you can again treat it with methanol, which will induce beta sheet formation again. So, the original silk that was water insoluble, through the lithium bromide treatment and through a series of processing steps has become water soluble, and then once it is processed, you can again induce the beta sheet formation, induce crystallinity through methanol treatment. All of it has to do with how these chemicals, reagents interact with the hydrogen bonds and how they disrupt the secondary structures in silk. I would encourage you to look up this process in more detail to get a better sense of what the exact nature of interactions are, how does lithium bromide disrupt the hydrogen bonds, and how does methanol induce crystallization in silk. The last polymer we will look at before we end this lecture is an example for what we would call a biological elastomer, which is elastin. And tropoelastin is the precursor for this, which contains both helices and alpha helices and beta sheets. And again, you see the familiar amino acids, glycine, valine, alanine, and proline. This is also a structural protein, so it is not surprising that 1/3 of the protein itself is made up of glycine. And the alanine and lysine rich segments lead to the helices. The glycine, proline, and valine rich segments lead to the beta sheets. Now, one important reason why elastin has elastomeric properties is because of this reaction that we have seen earlier between L-lysines with themselves or with unmodified lysine, which leads to covalently crossed networks. So, this decreases the solubility of the protein and or the fibers in water, but gives them elastomeric properties. So, shown here is a cartoon of that, where these represent the crosslinks, and these are the chains themselves, and you see when you stretch them, the elastin molecule uncoils to the extended conformation, which is this, and then when you relax, the chains recoil spontaneously and go back to the state. That is largely driven by entropic considerations. And this structural protein is very important in a lot of tissues where this kind of stretch and relax elastomeric behavior is very desirable. Some examples of which are shown here. One reason why your skin returns to its original position when pinched is because of elastin. Blood vessels return to their normal size after expansion because of elastin. Lungs are able to expand during breathing and go back to the normal size because of elastin. So, wherever you can think of tissues that require reversible deformation with elastomeric properties, most likely you will find elastin. Some of the concepts related to elastomers we have seen already in lecture 10. So, now would be a good time to stop and refresh some of those ideas. So, with that, I hope you get a good sense of some key aspects of protein structure property relationships, primary, secondary, tertiary and quaternary structures, the dihedral angles and the interactions that stabilize the different levels of protein structure, as well as structure property relationships in the context of collagen, silk fibroin and elastin, which we have seen in this lecture. Other classes of natural polymers will be the subject of the next lecture.